Thank you all for being here for my birthday. Yeah, this is one of the more memorable gifts that you can have for a birthday. And so I'm really delighted to be here and have the pleasure of being a recipient of the JapanPrize, one of the most important and prestigious prizes in the world. So it's very exciting, and I'm very honored to be among the members that have been previously selected as honorees for this amazing prize. And so today I'm going to share with you some different parts of the evolution of the field that I'm in, which is nuclear receptors. And this is an area that involves gene control mechanisms and developing new concepts over the last 40 or so years ago when we really got deep into this area. And so to get going on this, I'm going to get my timer here because I'm saying that it's off the node. I cannot. Okay, well, I'm just going to go, and I'll just, since my timer is not working, I can just go forever. Just kidding. But maybe if someone can give me a little hint here. All right, so it's 3:15. Okay, so I'm going to talk about nuclear receptors as it shows up there, and metabolic engineering and synthetic physiology. And so body physiology is going to be one of the core features. And they call it balancing the fat equation. And in this particular picture, you're seeing the transition of our DNA and the interaction with the food and nutrients that's coming in that goes into the tape measure of weight that we can be gaining if we start to expand our belt line. And you can see in this area that there's this interaction between the food and nutrients that we eat, how that's managed, and the role of the way in which DNA genetic networks play an important aspect in this process. And the nature of high-fat diets and obesity will be the underpinning of this talk. And so there's sugar and calories together are very classical ways to think about calories and nutrients, but at a molecular genetic level, it's really not what you think it is. And so this is in 2006. The New York Times actually ran a week-long front page series on obesity and diabetes and their associated complications. And so this is how big and important this area is. And our work and others have really focused on the central importance of nuclear receptors as in the heart of what you might call metabolic syndrome, and I'll talk to you about that in a little bit. This is a kind of a image or a way to think about body physiology. Body works in time and in rhythm and in space. It's three-dimensional, and everything that we're doing is happening in a circadian way this involves time and different dimensions. So we have to think about the way in which our body circuits work. These are, you can't really see these here, but these are the, I don't know if this works or not. But these are the basically 50 nuclear receptors that we have in our body. Every cell has at least 22 nuclear receptors. No two cells have the same combination. And all animals have nuclear receptors virtually the same that we have. And most of these, or many of these, respond to ligands or hormones or chemicals. and some are naturally active as stimulators, and some are repressors. And so there's a lot of features here. So we work and we live in something that's time, in rhythm, and in our environment. And the receptors control networks. I'll show you networks in a minute. And this is the mechanism for which nutrients can integrate physiologic pathways, behavior, and disease. And so we can look at this in a very simple way in the beginning, and then perhaps a little bit more detail as we go along. It's inactive, it's there, it's in place, but when you give ligand, the hormone is here, and the ligand activates the receptor complex, and these activation domains on the end make the gene come on, or they can make the gene come off. And we'll show you that a little bit more as we go along. And this is a little view of the Rosetta Stone. And some of you know the Rosetta Stone that was carved in 196 BC. And it was the inscription for Greek, Egyptian hieroglyphics and Demotic, and was found in the town of Rosetta in Egypt. And this essentially was the key to understanding these different kinds of languages. So for the different languages in the nuclear receptor family, getting the Rosetta Stone, which here I'm showing you, it's a little bit hard to see because these are the nuclear receptors. Up here, androgen receptor, estrogen receptor, progesterone receptor, vitamin A, vitamin D receptors, thyroid hormone receptors. These are the receptors. These are the DNA sequences that they will bind to. Every single cell in your body has 3.2 billion nucleotides. The receptor has to find the exact right spot within seconds, and it does that. And these are the DNA sequences that they will find in, very rapidly. And then there's the activators, which are their actual hormones, estrogens, androgens, and thyroid hormone, all the other components that are part of making the complete receptor. And so the Rosetta Stone basically is a way of thinking about how communication, how something that is inert, let's say a vitamin, when it interacts with the dynamics of the nuclear receptor, becomes dynamic. And so there's a lot of thinking to go behind that. And really, all of this relates to the evolution of our system. And speaking of evolution, there is one of the key thinkers in this is Charles Darwin. And we have the 150th anniversary of the of the origin of species. And in the origin of species, he spoke about the idea of survival of the fittest. And survival of the fittest, we kind of all know what that means, but part of the word fittest is fitness. And fitness is for animals moving around and being active. And so I think Darwin is really someone who's good to who was thinking about the origin of various aspects of the body. And I think it's very important for us to keep thinking about how that works. William Harvey is often called the father of physiology. And he, in 1867 or something like that, he wrote a book called De Motu de Cordis on the motion of the heart. And he was a pioneer. Everyone thought the liver at that time was the most important organ in the body. But in fact, he said the heart is a pump. It's a living pump that pumps blood around. This was like giving a telescope, to to look at the stars. It was like, this was an amazing advance, and how that worked, it was very important. And he created a science about the physiology of the body. And that led to Claude Bernard, another famous person in the field who came up with many key concepts such as homeostasis and the underpinning of survival and health. He called it the maintenance of the internal environment, the milieu or interior, is the basis of a free and independent life. If things are working together, it's good. But if they're not working together, then lots of things can happen. And so thinking out how they work, we have to think of how does vitamin A work? How does vitamin D work? How do all these different hormones work? And in that era, no one knew really. In fact, that was before hormones, I should say. But he really got the field going in terms of physiology and health. And following that, hormones were discovered between 1910 and 1920. Vitamin A, first vitamin. Vitamin D, the fourth vitamin. Thyroid hormone, not a vitamin, but these are all discovered, and they all had major awards that came with those because they were very important observations that these molecules that are simple can have complex impact on body physiology and health. And that leads from that era from 110 to 120, a void of trying to know how these hormones actually work. And it was the cloning of the glucocorticoid receptor that gave us the first real purchase on how these simple molecules, like thyroid hormone, which is a modified amino acid, a modified tyrosine, that is a very simple hormone, but it's one of the most important hormones in the body, and it activates so many aspects. And so simple things can be very complicated. For me, I wanted to understand that in detail, and that's where the cloning of the nuclear receptors was essentially important to be able to understand this process. And so if we look at the way in which Claude Bernard collected these together, it was organ physiology is the study of organic functions, including endocrinology, kinology, enzymology, nutrition, immunology, cytology, hemodynamics, and the amazing level of work that he's done. So this is the nuclear receptor superfamily as it developed, and it's in three parts. There was receptors for known hormones that are shown here. Another class, these are all the steroid hormones. These are the non-steroid hormones that were also part of it. These are ones that are all endocrine receptors. And then we found a collection of receptors that no one knew anything about. And this, reminds me of the idea when Howard Carter entered in 1922 King Tut's tomb. And in doing that, he found all these hidden treasures. He was the first one to get into the tomb. He was the first to see everything. It had not been touched by anyone else. I felt like with getting the glucocorticoid receptor, we unlocked a door and expecting one thing, we got exactly what we wanted, the glucocorticoid receptor. But when we walked into that room, there were all these other glistening things and there were other doors, and we knew that there was something much bigger than what we thought. And it's a little bit like I felt that way because the nuclear receptors then become the genomic key to unlock this door. And once we got in, we had a great time trying to understand the impact of these and how they really work. I'm going to talk about two of the receptors basically in the presentation. One's called FXR. It's a bile acid receptor. Mostly bile acids actually won a Nobel Prize for their discovery, but no one knew how they worked for another 70 years. Everyone thought they're just about solubilizing lipids to bring nutrients into the body, which they do. But in fact, they're potent hormones, and I'll show you that. And the discovery of the bile acid receptor in 1995 transformed bile acids to something really to another level. And then there's another nuclear receptor here called PPAR. These are the PPRs. There's 3 of these, and I'll talk to you about one of them. One of these stores and helps to store lipid. One helps to release lipid. One helps to burn lipid. And so there's different components of these ways in which these genetic networks interact with the nutrients and foods that we eat. So first I'm going to talk with you about is the PPRs. And this is just a little bit going back to this ring of physiology. And that you can deconstruct it in a visual way. And it's pretty hard to see in this complex, but this one single receptor, cloned by Ruth Yu and Kazu Misono, PNR, is the one single transcription factor that forms the eye. This is one single gene. It's a nuclear receptor superfamily member, not about hormones. It's about how you get vision. And there's one pathway, and it's PNR. And there are mutations in that and that people have with all sorts of vision issues and blindness. And so, and then there's receptors for sugar, fat, and salt. And so there's almost something for everybody, and I won't go through that other than to say you can put these together in different kinds of complexes. All of this is about reproduction. So moving ahead, because we now were able to get nuclear receptors, and we had the technology to get nuclear receptors, is now possible to take these new receptors that no one had seen before from these glittering prizes that we could get and develop new drugs. For many of these, we didn't know what they did at all. And so there was a lot of challenge of how do you figure out when you have something that is a nuclear receptor, you have no idea what the ligand is going to be. And so we can talk about that later, but it took a lot of effort and automation in our lab to create a library of drugs that we could just throw at the receptors. We had about 150,000 drugs in robotic machines trying to work these things out. But you can just kind of see these pathways, and basically every pharmaceutical company started to get into this business as well, and a lot of drugs have come out. About 25% of prescription drugs are nuclear receptors. So here is getting into the nature of metabolic syndrome. Obesity and the metabolic syndrome is shown here. When you're young and you're healthy and you're active, you look like the surfer guy. But as you get older, you're more sedentary, you're on a high-fat diet with poor exercise, you're now a channel surfer. And here's insulin resistance as a product of this, hypertension, hyperlipidemia, heart disease, and it's a very real challenge. And then along with that comes other problems of blindness, kidney failure, stroke, cancer. And I'm going to talk mostly about cancer and the nature of the impact of the nuclear receptors in disease mechanisms. And the thing about weight gain is it's actually rather easy. But as you see with Joe Lewis's, I went on a diet, swore off drinking and heavy eating, and in 14 days, I lost 2 weeks. And that's the reality, is that it's much easier to gain weight than it is to lose weight. And we were designed to, because we evolved with not a lot of nutrients in our environment, to store all the nutrients. And humans are very good at storing nutrients. And anyone in this room can last a month without nutrients as long as you get water. And we're really good at survival. And so I'm going to talk about two nuclear receptors that relate to what I call the in and out mechanism. And the two receptors here are the bile acid receptor, which I mentioned briefly before, and this PPAR delta receptor. And I call this outside in because the food that's coming into your body all goes through your digestive system and gets into the intestine. These bile acids come in during the meal from the gallbladder and the liver, and they solubilize the lipids, and they bring all the lipids and all the bile acids as chylomicrons back into the body. So everything that comes into the intestine goes right back into your bloodstream. And this is the single major master regulator of fat acquisition. And on the other hand, this is the inside out. This is the single most powerful receptor in burning fat. So one's going to be storing it, and the other is going to be burning it. And we made drugs for both of these receptors, and others have done that as well. And because this is about fat and fat storage, it's linked to fatty liver disease, heart failure, diabetes, NASH, GI diseases. I'll talk to you about that. And basically, the same with this is about fat burning and why this is so important and where it can play a role. And when we're looking at that currently in Duchenne muscular dystrophy, in heart failure, and lipodystrophy, in various aspects of diabetes. And so we're going to just move forward. And in this idea of inside out, we're going to create what actually the New York Times called this program that we started as exercise in a pill, and where we developed a specific ligand for PPAR delta, which was genetically linked to the fat burning program in the body. And a number of things burn fat. Exercise will burn fat. And when exercise burns fat, it's because PPAR delta is being activated. And it's being activated in part by the actual lipids themselves. So it binds long-chain fatty acids. PPAR delta binds like linolenic acid. Linolenic acid is about 20 to 25% daily of our diet. And some of the foods we eat are just activating these receptors. And it gives us a way to say, let's make a synthetic drug that has no calories and that we can control and see what happens. And I'll just take you through this a little bit. So what are the benefits of exercise? Because this is key to exercise, is burning energy. And the PPARs are the product of highly evolved complex genetic circuits. Again, it's all about lipids and small molecules activating these complicated circuits. And in the end, everything that we talk about for this is going to be circuits. But in terms of exercise, there's two ways to think of exercise. For some animals, exercise is specifically programmed. And for others, it's adaptive. So some humans can run long distances, long distance runners. Others can run fast. And so we're a hybrid species. And we can think about the nature of what causes a trait like that. What's the difference between a long distance runner or the fastest runner? And how does that relate to these circuits? One of the fastest runners in the jungle, this is one of my pictures, is a leopard cub, which is cute and elegant and shy. And it's one of Africa's most powerful predators. It can run 36 miles an hour very quickly, and there are very few animals that can actually beat that. And so it's designed to be fast, but it does not have long distance capacity. If you look at the spotted hyena in the lower left, this is total endurance. They work as a pack, and they drive the prey until they can't stop anymore, and then they attack. So these are super endurance. They're the best hunters in Africa. And then there are the bar-tailed godwit, which goes from Alaska to New Zealand or to Australia, depending on which, if you like, New Zealand or Australia. But they fly 6,000 miles in 9 days, and you can't train for that. So this is a genetically endowed trait. You can't just train to go 9,000 miles without stopping. And humans, on the other hand, are adaptive. We can train for being fast. We can train for being long distance runners. We're not hardwired in that sense. We have a mixture. And it's interesting, we can now break down some of the ways in which that mixture works. So one of my favorite ones that is the blackpoll warbler. The blackpoll warbler is another champion. So it's 5 inches long. It weighs about a little bit more than half an ounce. It flies to Alaska from Alaska to Cape Cod. And from Cape Cod, it eats as many bugs and little insects as it can, and then they all fly off together to South America. That's 90 hours. These little birds are flying 4 million beats of the wing. And again, no rest, no food, no water, over 2,300 miles. It's amazing how evolution and these networks work. They have a lot of PPR delta activity going on in that. And so we want to see how much we can take these observations and create potential therapies. And so it's like a prescription for endurance. And can a pill be a solution to the diabesity epidemic problem? And this is just one of the potential holy grails that one could think about as trying to use a drug that activates a genetic circuit to actually have the impact of that circuit, even and that is burning energy, even when you're not exercising. So once this starts the burning program, it releases and breaks down lipids, and just like when you run, the product of running is heat. And the product with the drug as well that we're developing is it breaks down the same lipids and you radiate it out as heat. And so Simon Cowell said, no, exercise in a pill is not, that's a loser. But we'll see what happens. And so these are back to the PPRs. This is the family that controls lipids, master regulators of lipids. This receptor, if you put it in any cell of the body or in a dish, it'll become a fat cell. So this is dedicated to making fat, storing fat. So this is the storage part. These are the ones that burn fat over here. And I'm going to talk about PPAR delta right there. And a drug called GW1516. This one that was made by Glaxo Wellcome. It's widely used by athletes. It's not a legally approved drug, but they use it anyway. It's very Russian athletes have been the most based on the vials that get thrown around in the Olympics. And then, so how does it work? If there is exercise in a pill, this is muscle that that we're looking at. I'm just going to kind of accelerate this. This is the nucleus of the cell. Remember, all cells have 3.2 billion nuclei, and this drug is going to be coming into the nucleus. We want to melt down some of the fat, get that fat burning. It's going to do it without the drug without exercise. Here, the drug is coming in. It's going to find all these sites, find where it's supposed to go on the 3.2 billion. Now the drug is coming in, and it's going to interact with the quiescent or quiet receptor and the repressor mechanism. This triggers a process which this repressor in green leaves. And now you can attract the activation mechanism that now allows this gene to be turned on. And literally, as fast as you saw it here is about how fast it goes in your body. This is not minutes. It's really a very, very rapid process. And this is happening throughout the genome. And when we say that it activates networks, there are thousands of genes in these networks that just get launched every time. So you really don't know when you just get a vitamin or some bile acids, what's really locked in that simple molecule can be a really amazing language that we didn't know about before. And I'm going to go here for the running man here. We're going to go from the cartoon to thinking about how we can actually begin to look at this the potential of this pathway. And we're going to use mice. Mice are really, they're not long distance runners, and they're really short distance. But what we have is two sets of mice. One that is not going to get the drug, the GW drug, one that will get the drug, which is here. We didn't really give them Diet Coke. That was just a joke. But we did give this one a cocktail. Just kidding. And so we're going to put them on a treadmill, and then we're going to see how they perform. And there is the mouse treadmill. It's kind of like a human treadmill, except they can't get out. They're going to run until they cannot run anymore. And one of these pairs of mice has the drug, and the other doesn't. I'm going to skip ahead here, try to keep this going a bit. And what you're going to see, shortly, is that one of them is going to give up, and because it can't run anymore right there. This one will go an hour more without stopping. That's just, was really exciting for us to see that giving this drug could actually change, without any training in that mouse, its ability to run a longer distance. Part of it shows that if you get the drug is pre-training your myofibers in the muscle to be ready to run a long distance. And whereas the vehicle, the mice don't want to run long distance. They want to run short, fast distances. And so this just is, we now can technically control part of this running and endurance pathway. And if you do this with a series of animals, basically what you can see is that the total running time here, these are minutes of the normal mouse and the one that gets the drug, about 100% increase, doubling the total time and the total distance. And this is the beginning of the era of making newer and more refined compounds. So Simon Cowell changed his mind. This is actually potentially pretty good. More recently, a startup company that we have in Boston, we developed the next round of PPAR delta drugs that are very uniquely designed with a special chemistry, and they're very safe, and they do not activate thousands of genes. This is the surprise in making this drug. It activates a very small set of genes. And so within these networks, there are sub-networks. We targeted the sub-network, and this is just it's not so easy to see, but the control mice are here. The mice that get the drug is called MA here. This TR means we're training the mice. These don't get trained, but we'll take normal mice and train them. And then they only have one pair of mice or one set that has the training and the drug together. Point is that this is the normal run for a mouse. It's a little bit like 180 minutes or so. Then if you use the drug by itself with no training, the drug by itself boosts the running distance about close to double. And these are the mice that they were kind of run there. They kind of run a different length. If you just do training, training also works better than control. So obviously when you train, you're going to get better, but you're not as good as just the drug alone. But if you combine the drug and training together, you now get this. And what is really surprising, is a little bit hard to see, is the champion of this. And that's this mouse here. My postdoc went into the lab. You have to watch these, every animal run until it's exhausted. And he went in at 3 p.m. And at 6 p.m., he called his wife and said, just have dinner. At 9 p.m., he said, put the kids to bed. At midnight, he says, I'm running late. Just go to bed. And the mouse ran just short of 10 continuous hours. So the combination here shows, this is why we call this super physiology. There's no mouse that we know on the planet that can run that long and be happy at it at the end. And so this just shows you the potential of discovery and how you can begin to deconstruct it, both in the mechanism, but what you might be able to achieve in the context of what we know are the properties of the body physiology, in this case, exercise and endurance. These are all the benefits. You get a lot of benefits from even moderate exercise. Walking will lower your sugar levels. Any kind of exercise is really good for you. But oddly enough, this is the one without exercise, but it's the PPR delta. And that is giving us all the benefits, basically, that we're seeing with metabolic fitness. And I was told by the most top people in exercise physiology that there will never be a drug that gives you the benefit of real exercise. And I go, what is real exercise? Can you define that for me? Is that like dancing? Is that weightlifting? Is that ballet? Is it a football player? Exercise is actually everything. We have to think about these things that the word doesn't actually always express what it really means. But in this case, PPR gamma is activating these same networks that we think are very important. We're doing a lot of work now in aging, in Alzheimer's, in MS, and lots of work in the CNS. And so looking to see how this can develop. And actually, when you run, you increase memory cells that are being created. And so does the PPR delta drug on its own, even in absence of that. So I won't go through all these benefits that you get for metabolic disease and obesity, the neurodegeneration components with Alzheimer's and Huntington's, vascular problems. It's really very good in the heart as well. And then these aging risks that link to obesity, high blood pressure, heart disease, frailty, primary muscle myopathy, or chronic fatigue syndrome. So this tells us that we need to continue pursuing this in a way that can be potentially helpful to people who really can't get the needed exercise that they need. And I will tell you personally that I think it's this obesity epidemic is also part of an exercise non-epidemic. And part of that is also linked to modern technology. The kids are now getting iPads when they're 1 or 2 or 3 years old. Kids grow up with playing games instead of running around. And computers are designed in their programs to keep you at the computer. And that means these boys that would normally run around and create chaos are now making the chaos, but on a computer screen, so you're not burning any energy. And so we need to think about the impact of technology on things like our physiology. And just to get towards the last part of this, and I think I'm getting beyond the last part. So we have made a new drug to the bile acid receptor FXR, which is shown here. This is a drug that is oral. It does not get into the body. So you take it orally, then give it to the mice. It goes through the stomach, the intestine, goes all the way through. It activates this master regulator FXR, just like regular food would activate that. So we call this the meal in a pill. It's activating this network, but there are no calories that are coming in. But all of the hormones and the regulators of body physiology are being released. Lots of GLP-1, which is one of these key hormones for the weight loss drugs. And so we can now activate many of these pathways in a way in which the drug doesn't even stay in the body. It goes through this network and comes out. And I'm going to just show you just a little bit about what it can do. And in the context of cancer, I'm just going to show you a little link here with a mouse model for colon cancer. And the thing about looking at the disease mechanisms for colon cancer, the most common one is Adenomys polyposis coli. APC-min is the most common mutation for colon cancer. About 80% of people that get colon cancer have this mutation. Our mice have this mutation. And just to show you the bile acid pool in a normal mouse on a normal diet is whatever this number is. Let's say it's 2, and we can measure the serum bile acid pool in these animals. If you have the APC-min mutation by itself, what you get is a much larger bile acid pool, and so the mutation is acting not so much as a oncogene, it's stimulating some feature of this oncogene that we didn't know anything about. And people did not know what this did, but it actually controls bile acids. When you give that drug, that FXR drug, that brings it down to this point in the context of this particular mouse. If we then do basically the same experiment, but on a high-fat diet, this is on a normal diet, then we're going to go to high-fat diet, you can see that the serum bile acid is gigantic. I'm just going to show you in the mouse that has the mutation and that is bringing in a lot of inflammatory action here at super high levels. This is 10, but this is 200. That the bile acids are going crazy. But when you give the drug, FXR, you bring it back to basically close to normal. So we're overcoming virtually the full effect of diet and genes. And so this is where these genes and the diet come together to think about these mechanisms. And I'm going to just go back. I'm going to go to the end just the acknowledgments to thank my family. My wife and daughter are here today, and my colleagues who over the years and decades have been amazing, and we couldn't do anything without such talented people. And those who are involved in the current research that I was talking about, It's over here. This is my leadership who oversees a lot of the work that gets done. I want to acknowledge my former postdocs, Kira Kakuzuka, who's in the audience here today. Thank you. Hiro Ogura, Hiko Ogura. He was here yesterday for the award ceremony. And Satoshi Ogawa, and I also want to mention Kazu Musono and Ruth Yu. And these are really amazing people that had been in my lab and helped pioneer all these programs and take it to the next level. And really, it's all about, in running a lab, the next generation. And if we can turn this over to the right people, I think we'll be in good hands. And with that, I'm going to stop. Thank all of you very much.